The Biophysical Society Lecture is the Society's highest honor, recognizing work that has fundamentally shaped how biophysics is done. We are joined now by Louis Kay, whose development of modern solution NMR methods has transformed how scientists study the motions of biomolecules, not just their structures. Thank you so much for being here.
Thanks for having me.
I want to start off with a lot of these structural biology was always focused on static structures. Now that we know that proteins keep moving, what's changed?
Well, it's actually an interesting question. You know, going back to the days when crystallography was just evolving, so some of the pioneers in structural biology, Max Perutz, for example, were producing three-dimensional images of protein molecules. These were very static, and people were adamant that these protein molecules did not kick or scream or dance. But in terms of what's changed, I think there was the realization that proteins were dynamic, that you couldn't just have static entities that were performing function. The question was, how would you actually study these? But I think the dynamics of molecules were actually appreciated much earlier on than we think. It's just that people were focused on doing other things rather than on perhaps what NMR should have been focused on from the get-go, which is elucidating function through an understanding of molecular dynamics.
And now that we can project structures computationally, how does that change the experiment solutions like NMR?
Well, it means that NMR spectroscopists, by and large, don't have to focus on doing structures of proteins or biomolecules anymore, because we can generate those structures. Now, I should say that the fact that you can generate those structures computationally doesn't mean necessarily that you're generating the right structures computationally. I mean, these computer programs are wonderful, but they've been trained on a set of structures and not trained on other ones, and it remains to be seen how well they're going to do on the structures that they're not trained on. I suspect very well in most cases. But what you have to understand is you can't really talk about a structure of a protein. So imagine a protein sort of like a person. You have a photograph of a person. That's a structure of the person, but it doesn't tell you how the person works. It doesn't tell you how the person interacts with his or her environment. So it doesn't tell you about the dynamics. You get a structure, but you don't have higher energy structures of the molecule, if you like. And these higher energy structures, which occurs simply because molecules are bumping into other molecules all the time, can create changes in the overall three-dimensional structure of the molecule in ways that allow different functions to occur. So you have the sort of ground state energy, the low energy of the molecule, and then you have higher energies. Just like if I were to take a punching bag, right? The ground state of that punching bag is just sitting there. And then I punch the punching bag and I watch how it responds, right? That's what we want to be able to do. Now, we have to know what a punching bag looks like in the first place, so we need structures. But we need to understand how those structures evolve as a function of time.
And talking about new tools, did you ever think during your research that your methods would make such an impact in the field?
Well, my methods and everybody else's methods, I mean, this is not a single person enterprise for sure, as no sort of advance in science is. But no, I never thought that people would be actually using these methods. I mean, as a postdoc in collaboration with Dennis Torsha and Ad Bax at the NIH, you know, two great NMR spectroscopists, you know, there was the development of methods, again, that were spurred on by molecular biology, for which we could produce proteins that were labeled in ways that we could now have access to every amino acid in the protein and ask how it moves, how it wiggles and waggles, what the time scales are. And that was very exciting, but I never thought that it would catch on. I never thought that some of my most well-cited papers would be from those sorts of ideas. No clue.
Well, your passion is definitely having an impact, so thank you for what you do.
Well, thank you for your interest.
And thanks for being here.
Super.